In this video, we're going to be discussing Chapter 3. Chapter 3 will go over prokaryotic cells, their morphology, structure, and function. All right, so to get into some detail about the prokaryotes, when we think the term pro, we think before, and then kary means nucleus. So prokaryotes are missing the membrane-bound organelles. They don't have a nucleus, but what we can see are that they do have ribosomes, and ribosomes are going to be found within the cell, and the ribosomes are going to have a function to actually help to make proteins. We're also going to see that they do have a cell membrane, and a cell membrane is going to be that selectively permeable membrane. So it's going to keep things from basically staying in and out of the cell or really help with the control in and out of the cell. We'll notice that there's a cytoplasm. They do actually have DNA, but it's just not found in a nucleus. Instead, we'll see it found in a nucleoid. Now, we'll see the cell membrane, and then there will be a cell wall on the outermost portion of the cell. And for prokaryotes, especially bacteria, their cell wall is composed of something called peptidoglycan. They may also have another layer called a capsule or glycocalyx that's going to help from preventing the cell from drying out. It also helps to establish some antibiotic resistance and even resistance against our white blood cells, against our immune system. We'll also see inclusion bodies as well, and those inclusion bodies can differentiate and vary depending on the prokaryote. And we'll also see that they have a flagellum, which is a tail, which is going to be used for locomotion. Here are just a few things that we can see on this picture here. We'll get into more detail about those specific structures here in just a moment. All right, so I had made mention of the cell membrane and the cytoplasm. When we think about the cell membrane is going to be what we call selectively permeable. And the reason that it's selectively permeable is because it has this phospholipid bilayer. It's two layers of phospholipids. The phospholipids are going to have these hydrophilic heads, meaning that they like to interact with water, and they'll have these hydrophobic tails, meaning that they don't like to interact with water. And so this creates what we call a phospholipid bilayer. So the heads will be on the outermost portion, the tails will be on the inside. Now, this creates what we call a fluid mosaic model, and this is basically describing the understanding of how we understand the membrane structure. With the phospholipid bilayers, there's some proteins that are going to be added in as well. Some of those proteins will be on one side of the cell surface. Some will actually be embedded through the entire part of the cell membrane. We'll also see some carbohydrates, and we'll see some glycolipids found within the structure. And on top of that, we may also find cholesterol as well. All right, so here's just a picture of that cell membrane. So again, we discussed the cell membrane as being selectively permeable, meaning that some things can go in and some things can go out. But when we look at this picture, we're going to see that we have this phospholipid bilayer. So we have these hydrophilic heads on one side, and we'll see them on the other side as well. Let me draw a picture here. So we have the phospholipid bilayer, and we have the heads on one side. The tails are going to be in the middle that we're highlighting in red. And we'll also see these proteins. So again, some proteins will actually go through the entire cell membrane. These are what we refer to as integral proteins. And then there are some peripheral proteins that are going to be on one side or the other. We also may see, again, some carbohydrates and some cholesterol. So that's not really shown here at this point. The major key here is to see that phospholipid bilayer. And so again, the heads are going to be hydrophilic, meaning that they like to interact with water. The tails are hydrophobic, meaning they do not want to interact with water. And that's why they're actually in this arrangement to really protect those tails from associating with water. All right, so when we think about the functions of the cell membrane, again, it creates this selectively permeable barrier. So again, only a few things can pass freely through. And then when we think about the cell membrane, on the inside, it's going to contain the cell contents. Now, what also can happen too is we have some transport proteins that can allow for things to move in and out of the cell using a concentration gradient, and it will also utilize energy as well. But the cell membrane is also going to be very important when we talk about energy generation in terms of the electron transport chain. And we'll discuss that a little bit more later on when it comes to actually thinking about energy production. All right, so thinking about inside of the cell, what would we be looking at? Again, the first thing that we'll talk about is the cytoplasm. The cytoplasm is going to be a liquid that actually is enclosed by the cell membrane. And so it's going to be like a more viscous or thick fluid that we'll see. And most of the biological functions are necessary in the cytoplasm. These are all necessary for survival and actually being able to maintain day to day life functions. All right, so what we will find in a prokaryotic cell is DNA. Again, for prokaryotes, they do not have a nucleus to house the DNA, but rather they have what's known as a nucleoid. But the DNA that's found in there is going to be single and it's going to be a circular chromosome. This is different than eukaryotes. Now, they also may have an additional element or additional chromosome that's known as a plasmid. It's usually a very small circular molecule, and this will have additional genes, but those genes are not needed for survival or for growth. However, they do help to kind of create kind of a incentive, if you will, or they just kind of give a one up, if you will. Usually, they're going to be very helpful for carrying antibiotic resistant genes. So, while it's not essential for growth, it does give them sort of that leg up in comparison to others. All right, we are going to find ribosomes. And ribosomes are going to look like little dots, if you will. They are involved in protein synthesis. And so, what they're going to do is they're going to help to join amino acids together, and they're going to help to form these peptides or these proteins. And each ribosome is composed of a small and a larger subunit. It's indicated by an S, and that reflects how fast they settle. The faster they move down toward the bottom, the higher the S value and the greater the density. So eukaryotes versus prokaryote, when in terms of ribosomes, are going to be different. Prokaryotic ribosomes are what we consider to be a 70S, and they're composed of 30S and 50S. Now, as you can see, it doesn't total up to be that number, so how they get that number is a little bit different. But eukaryotic ribosomes are going to be an 80S. All right, we're going to give a little bit of background, and we're going to talk about the kind of the origin of mitochondria and chloroplasts for just a hot minute. So this is going to be what we know as the endosymbiotic theory. And the idea is that ancestors of mitochondria and chloroplasts were actually once bacteria. So mitochondria and chloroplasts are found in eukaryotic cells. But they were believed at one point to be bacteria that were just residing in other cells, and they were sharing this beneficial partnership. So the mitochondria is the powerhouse of eukaryotic cells, and it's the site of the electron transport chain or the electron transport system. And chloroplasts are extremely important for plant cells to allow for that photosynthetic detail and photosynthesis to take place. So while these guys are eukaryotic organelles, they were once thought to be bacteria. Right. And then when we talk about this sort of evidence behind the theory, they have genes that make 70S ribosomes. And remember, eukaryotic cells are going to be an 80S. So the DNA comparisons allow for that kind of idea of this close relationship and how they've also shown close relationships to obligate intracellular parasites. They also elongate and divide similar to bacteria, and they also have a double membrane. So this is why, again, it kind of helps to sort of move that way towards this evidence that mitochondria and chloroplasts were at one point bacteria. But if you want to know more information, I do have a YouTube video linked below so you can get into a little bit more detail behind it. All right, and then finally, we're going to talk about inclusions. So inclusions can be found in not all bacteria, but some bacteria. And this is going to have little granules inside of them, so it allows for storage. So they can store starch or glycogen, whatever they need for survival.